Welcome to this overview of the history of bioinformatics, where we trace how computing and biology came together to transform science. In this presentation, we'll explore early milestones, key technologies, important databases, and future directions. And the slide image hints at the humble computing setups that started it all and laid the groundwork for today's advances. This slide outlines what we'll cover: the origins of bioinformatics, the rise of sequence databases, genomics, and high-throughput data, core software and algorithms, real-world applications, future trends, and a concise summary. Think of this as our roadmap, guiding you through historical context, technical breakthroughs, and the practical impact that links computation to biological discovery. To begin our first section, we'll look at the origins of bioinformatics and how early computational thinking entered biology. This part sets the scene by explaining why researchers first turned to computers, how mathematical ideas influenced analysis, and why those early steps were crucial in shaping the tools and questions that drive modern computational biology. Early computational biology began when scientists started using computers to analyze DNA sequences, predict protein structures, and manage genetic data. Those first algorithms for comparing sequences and the influence of mathematics and statistics provided the analytical backbone, enabling pattern recognition and evolutionary insights that turned experimental observations into digital data we could study at scale. This timeline highlights major milestones that propelled bioinformatics forward, from the discovery of DNA's double helix to formulation of the central dogma and the earliest gene sequencing efforts. Each milestone acted as a technological or conceptual springboard, allowing researchers to translate molecular findings into computational problems and, ultimately, to develop tools for large-scale biological data analysis. Moving into the next section, we'll examine how sequence databases emerged and why they mattered. Databases turned isolated sequence files into shared resources, enabling global collaboration and reproducible research. In the following slides, we'll explore key repositories, the algorithms that leverage them, and the cultural shift toward open data that accelerated discovery. The creation of biological databases like GenBank, EMBL, and curated protein resources transformed how sequence data was stored and accessed, enabling researchers worldwide to share nucleotide and protein sequences. These platforms standardized data formats, accelerated discovery through broader access, and underscored how collaboration and data sharing became essential for progress in bioinformatics. Sequence alignment tools revolutionized how we compare biological sequences, starting with dynamic programming approaches like Needleman-Wunsch, and later fast heuristic search tools such as FASTA and BLAST. These methods made it practical to search massive databases, enabling comparative genomics, evolutionary studies, and rapid identification of similar sequences across species, which greatly boosted research efficiency. Now we transition to genomics and the era of high-throughput data, where sequencing moved from individual genes to entire genomes. This section will cover the technological breakthroughs and computational challenges introduced by large-scale projects, and how new sequencing methods and pipelines reshaped biological research by producing unprecedented volumes of data. The Human Genome Project aimed to map the entire human DNA sequence and fundamentally change genomics by driving automated sequencing, computational assembly, and large-scale data analysis. It also exposed data integration challenges, including diverse formats and the need for accurate, shareable datasets, and set standards that enabled subsequent genomic projects and bioinformatics infrastructure. Next-generation sequencing transformed genomics by enabling massively parallel sequencing that increased speed and reduced cost, creating a data explosion that demanded new storage and analysis solutions. This slide highlights how these technologies spurred cloud computing, advanced compression, and scalable bioinformatics pipelines to process, align, and interpret vast genomic datasets efficiently. In this section, we'll focus on bioinformatics software and algorithms, the tools that turn raw sequence data into biological insight. From annotation and assembly tools to structural prediction and machine learning methods, these algorithms form the practical core of modern bioinformatics, automating complex tasks and enabling discovery across many biological domains. Sequence analysis software includes gene prediction tools that identify coding regions, protein structure prediction methods that infer three-dimensional shapes, and functional annotation systems that assign biological meaning. Together, these tools help researchers annotate genomes, prioritize experimental targets, and interpret sequence data in the context of molecular function and biological pathways. Machine learning has become central to bioinformatics by improving pattern recognition and enabling data-driven predictions. It supports genomics and proteomics through models that predict gene expression, identify functional elements, and infer protein structure and function. And recent AI-driven advances have substantially improved accuracy and automation in many bioinformatics workflows. This upcoming section highlights the wide-ranging applications and societal impact of bioinformatics, from drug discovery to agriculture and environmental science. We'll look at how computational methods enable targeted research, help personalize medicine, and support sustainable practices, illustrating how bioinformatics translates complex data into real-world solutions. In biomedical research and drug discovery, bioinformatics drives target identification by analyzing biological datasets to find disease-related genes and proteins, supports personalized medicine by tailoring treatments to genetic profiles, and accelerates biomarker discovery for early diagnosis and targeted therapies, thereby improving both development pipelines and patient care. Bioinformatics also plays a major role in agriculture and environmental science by enabling crop genome analysis to breed resilient varieties, by supporting microbiome studies that improve soil health and productivity, and by guiding conservation genetics efforts to preserve biodiversity and manage endangered populations using genomic insights. Now we shift to future directions in bioinformatics, where integrating diverse data types and emerging technologies will define the field's next phase. This section previews trends like multi-omics integration, AI-driven analysis, real-time sequencing, and cloud-based collaboration that promise deeper biological understanding and faster, more flexible research workflows. Integrating multi-omics data presents major challenges, including storage, standardization, and computational scaling, but it also enables systems biology approaches that model complex interactions across molecular layers. Predictive modeling that combines genomics, transcriptomics, proteomics, and metabolomics can improve disease prognosis and personalized treatment by revealing hidden molecular patterns and mechanisms. Emerging technologies such as advanced artificial intelligence applications, real-time sequencing, and cloud-based collaborative platforms are reshaping bioinformatics. These innovations enhance predictive modeling, enable immediate data-driven responses, and lower barriers to global collaboration, together creating an ecosystem where data is more accessible and analyses are faster and more powerful. We've now reached the final section, a summary and conclusion that ties together the history, tools, applications, and future possibilities of bioinformatics. In closing, we'll recap the major developments and reflect on how ongoing technological progress continues to expand what's possible at the intersection of biology and computation. Recapping key historical developments, bioinformatics evolved through pivotal milestones like DNA sequencing and large-scale projects that demanded new computational tools. And continuous technological evolution has transformed the field from simple analyses into complex, integrated workflows, setting the stage for ongoing innovation and deeper biological insight. Finally, bioinformatics remains essential to modern biology by enabling large-scale data analysis, accelerating discoveries, and integrating diverse biological information to support genomics, proteomics, and personalized medicine, while also facing challenges such as data privacy, integration complexity, and growing computational demands that will shape its responsible future development.